Mendelian inheritance refers to the patterns of inheritance that are characteristic of organisms that reproduce sexually. Molecular genetics refers to a branch of genetics that deals with the structure and function of genes at a molecular level. GMOs are created when organisms whose genomes have been engineered in the lab in order to favor the expression of the desired physical traits or the generation of desired biological products. How they all tie in together. On the molecular side, recombinant DNA techniques are used to manipulate genetic information within and between plants, animals, and microorganisms in order to produce genetically modified organisms. Typically, GMOs are used to improve food in the size, taste, and appearance. On the Mendelian side, if the crops were left alone, they would adhere to Mendel's laws of segregation, independent assortment, and dominance. Take a look at the strawberries. The one on the left has been genetically modified to grow larger, and the one on the right hasn't been modified at all and is small in nature.
The basics of GMOs. On this slide, I'll be talking about genetically modified organisms, more specifically genetically modified crops. Scientists and farmers do grow genetically modified crops for the last 20 years or so. The reason why they would decide to grow genetically modified crops is because that produce or crop might be in high demand, or it might take longer for it to yield. And also another benefit of using genetically modified crops is that it reduces the use of pesticides. The pesticides are used to keep pests away, like insects, bugs that might carry diseases and kill potential crops. So genetically modified crops can be grown and engineered in a way that prevents insects and other pests to penetrate, eat, infect the crops that are being grown. So the things that they look for, there's different types of genes, just like in humans. There's different types of genes in crops, such as size, color, how much a plant will yield. And so they typically try to cater to the more voluminous and higher yield products that can grow better and bigger. Just as a consumer, that's what we would like to see. So as farmers and scientists, that's what they try to grow and produce with these genetically modified crops. And in addition to all those benefits of getting a bigger yield of the product in size, color, and maybe even improve taste, you can also engineer it to survive certain weather conditions. The types of genetically modified crops that we grow all over the United States is like cotton, soybeans, corn, apple, potatoes, canola, papaya, and squash, because those are in such high demand. And there's certain traits that we do desire about this produce, and we do need a lot of it to keep up with our population. Scientists do look into what's the best way to get as much as you can out of a plant. And in order to do that, they have to look at the genes inside the plant. And with the use of biotechnology, such as transgenetics and gene editing and using the CRISPR, it helps produce higher yield produce. So where did GMOs start? I will continue on the next slide. How did Mendel help us develop GMOs? So through Mendel's pea plant experiment, the technique that he used to crossbreed the pea plants is now known as traditional crossbreeding. He saw that there is different traits in each parent plant, and by crossbreeding each plant, it yielded a different combination of traits. And so through that, that helped scientists take that technique and see how maybe in taking these physical traits and crossbreeding it with another desirable physical trait from a different parent plant and how they can get the most desirable produce out of crossbreeding the parent plants. So a concept was taken out of the pea plant experiment called inheritance of traits, which essentially what I just explained is taking a trait from a parent plant and it's getting passed down to the offspring. So through this experiment, we were able to see the direct impact of traditional crossbreeding and seeing maybe if there's a desirable trait from one parent plant and another desirable trait from different parent plant. If we crossbreed them together, which one will come out dominant, which also helps figure out if it's a heterozygote trait or homozygote trait. And that helped us segue into the beginnings of the studies of genetically modified traits. But through more research and development of biotechnology, there has been more techniques developed that can actually go into the DNA of the parent plants and really pick out what desirable trait, rather than doing the traditional crossbreeding that Mendelian genetics has been dealing with, and more so looking where Mendelian genetics was more so looking at heterozygous traits and homozygous traits. You can actually, with these new techniques like transgenetics and mutagenesis and the CRISPR gene, you can actually pinpoint what genes you like and take them out and put it into a DNA strand and produce the more desirable produce, which in the next slide we'll be getting into more transgenetics, moving out of Mendelian genetics.
Now, to better understand how to modify the flavor profile of the product in order to make it more desirable and make it able to resist different elements of the climate, let's go to the transgenic side, meaning the genetics at the molecular level. Indeed, today, the creation of GMOs is mainly used in agriculture to make a plant resistant to herbicide or to modify it so that it produces an insecticide itself or both. To do this, the DNA of the organism in question is shaped or modified to endow it with properties that it did not possess naturally. This can also have an influence on the aesthetics to make the product more attractive to the consumer. The question remains how the genetic information of the plant can be modified. In order to genetically modify an organism, one must have access to its DNA. This will then allow access to the genes that contain information about the characteristics of living organisms. The technique used to obtain GMOs is called transgenesis. It consists in introducing a foreign gene, called transgene, in the genome of an organism. A transgenic plant is therefore a plant whose genome has been modified by the introduction of a gene, which can come from another plant, a bacterium, or any other organism. This gene can code for an enzyme that is involved in the ripening of fruits, a substance that blocks the multiplication of a virus, a new protein, for example a toxic compound for insect pests. After locating an interesting trait in another living organism and identifying the protein responsible for this trait, we identify and isolate the gene encoding for this protein. A gene construct is made containing the gene of interest and the DNA sequences, promoter-terminator, essential for its functions in the genome of a plant cell. These sequences are involved in the regulation of a gene expression. They make it possible to target the place of expression of the gene in the plant, or even to ensure that it is only expressed at the necessary moment during insect attack or infection by a virus, for example. This gene construct is then inserted into a bacterial plasmid to be multiplied and introduced into the plant cell genome using two methods. On one hand, the biological transfer, which consists in using a vector, and on the other hand, the mechanical transfer characterized by the gene construct carried by tungsten microbeads, which are projected into the plant cell. Then we select the cells expressing the added gene, and we regenerate whole plants from these cells. The plants are then tested for conformation, development, stability, and transmission. In general, in order to integrate new genes into an organism, there are different methods categorized into two parts, direct and indirect. Among the direct transfer techniques, there are electroporation, which consists in subjecting a mixture of protoplast and DNA to electric shocks. The electric field causes the destabilization of the plasma membrane of the protoplast and leads to the opening of the membrane pores, thus facilitating the passage of DNA into the nucleus. There is also microinjection, which consists in introducing the foreign gene directly into the cell to be modified using a micromanipulator mounted with a microscope. The protoplast to be transformed is held with a microneedle, and the gene with its promoter-terminator complex is introduced into the nucleus with a micropipette. Finally, there is biolistics, the most common method, which consists in propelling the transgene into the plant cells. DNA-coated metal microbits are projected at high speed onto the cells to be transformed in order to cross the wall and integrate their genome. Among the indirect transfer techniques, there are biological transfection, which is a more natural process based on the use of the properties of plant pathogenic bacteria. The gene introduced in a plasmid thanks to the different enzymes is then transferred in the bacteria before being integrated in the nucleus of the plant cell. Then it will be integrated in the genome. There is also lipotransfection, which aims at trapping the gene of interest in the liposome, a spherical structure made of lipids. The latter have the ability to fuse with the membrane of protoplasts to release their content, in this case the gene of interest, into the protoplast cytoplasm.
Examples of GMOs include cotton candy grapes, tomatoes, corn, and potatoes. Horticulturist Dr. David Kane and his team at Grapery changed the DNA in regular grapes to create a cotton candy taste. Dr. Kane says he breeds seedless grapes, but that they can't reproduce on their own. First, male grape flower pollen is extracted, then carefully brushed onto the female clusters of the target plant. After fertilizing, baby embryos are taken out and grown in test tubes one by one before they are ever planted in the field. Dr. Kane created almost 100,000 test tube plants before he came across the cotton candy flavored grapes. He says the whole process takes anywhere from 6 to 15 years, so he and his team may be waiting, replanting, and redoing the whole process all over again for a while. I've never tasted these grapes before, but according to Dr. Kane, they taste exactly like cotton candy, but without the stickiness or calories. Dr. Kane and his team have to widen the plant's gene pool and mix in genes from less common grape species to get a vanilla flavor into the regular grapes naturally. The extra hint of vanilla is what gives the grape that extra hint of sweetness. Some bacteria have a trait that allows them to produce a chemical that harms insects. That gene can then be taken out of the bacteria and inserted into the DNA of a tomato. The tomato will then start producing the same chemical as a bacteria to resist insects. Tomatoes can now produce their own pesticide, as well as preventing them from getting mushy or being able to produce more sugars. One method used to create this GMO is a gene gun. It shoots new DNA into the cells of the tomato. The original DNA will spread apart as the new DNA inserts itself. This specific DNA change will not change the tomato's appearance, but it will help farmers limit the amount of money spent and decrease the amount of pesticides released into the environment. Corn's wild ancestor is a grass called teosinte. Teosinte doesn't look like maize, especially when you compare its kernels to those of corn. But at the DNA level, the two are very alike. They have the same number of chromosomes and a similar arrangement of genes. Teosinte can crossbreed with modern maize varieties to form teosinte maize hybrids that can go on to reproduce naturally. Scientists can study those hybrids and their offspring through the process of genetic archaeology. This process helps geneticists understand what is happening at the DNA level to make teosinte and maize so different. They have calculated that only about 5 genes were responsible for the most notable differences between teosinte and maize. In more recent years, geneticists have used advanced molecular biology tools to pinpoint the roles of some of the genes with large effects, as well as many other regions across the genome that have subtle effects on maize domestication. Most common effects include types and amounts of starch production, ability to grow in different climates and types of soil, length and number of kernel rows, kernel size, shape and color, and resistance to pests. Potatoes have a gene that causes them to bruise when damaged. New GMO potatoes silence that gene so it cannot be expressed. The potato still gets damaged, but the symptoms are hidden. They have also been altered to produce lower levels of acrylamide when cooked. Acrylamide is a chemical that can form in some foods during high-temperature cooking processes, such as frying, roasting, and baking. Acrylamide in food forms from sugars and an amino acid that are naturally present. GMO potatoes have been engineered through a method of gene silencing called RNA interference, also known as RNAi. This genetic engineering technique results in a potato that hides the symptoms of black spot bruising rather than preventing it. First, mRNA carries genetic instructions from the cell nucleus out to other parts of a cell. RNAi then begins when a dsRNA is placed inside a cell. The dsRNA gets cut up by enzymes, paired with proteins, and then ends up binding to a specific target site where it fits on the mRNA. This can prevent the mRNA from delivering all of its instructions, effectively silencing the desired gene. In conclusion, Mendel's knowledge and experiment with pea plants helped segue into a giant industry of GMOs with the use of molecular genetics.